Bismillah. Assalamu alaikum wa rahmatullahi wa barakatuh. My name is Umar. Today I'll be presenting on my topic called Painting with Photons, Light Cells and the New Art of Cancer Detection. I'm hoping to show you that the title, The New Art, isn't just figurative, but it's quite literal. Like I'll be showing you some really beautiful paintings where I'm trying to model biology as a subject, but using physics as a paintbrush. Unfortunately, like all dry academics, I start off my presentation with an agenda. I've oriented the whole thing around six really big questions. One, what is the problem the doctors are facing that I'm trying to solve? Two, what is the question that I'm trying to answer? In like what specifically scope down, what is the solution? Three, what experiment am I going to run to try and solve or answer that question? Four, did that experiment work? Was I able to get results? Five, were the results meaningful anyway? Like, was it just beautiful data or was it useful for doctors in any sense? Thanks. And six, now that I've run that whole experiment, what was the point? Where do I go from there? To start off the presentation, I want to talk about the art of doing cancer detection as it stands today, like the gold standard. It's good that we had a presentation on CT, or at least using CT, because that studies the macro anatomy or the gross anatomy. I'm using microscopy, so just a normal microscope, you're putting something under and you're looking at it. And that's used to study the microanatomy of tissue, looking at like cells, organelles, things like that. This here is a needle core biopsy of breast tissue from a patient in Alberta. Biopsies are like the go-to way to diagnose cancer. It's how doctors, if they're suspecting that a mass is tumorous, it's how they confirm that. Or if they already confirmed it, it's how they investigate the pathology to then go on to figure out what's the best clinical intervention. On the right-hand side is an annotated version of that. This basically tells you where in this image is bad and where it isn't. So everything that's red is tumorous, and then the blue or green is different types of benign tissue. This is the go-to way. I want you to kind of look at the red section and then look at the corresponding section here and see if you can figure out, like, you can independently figure out what's cancerous and what's not. It's really tough. Like med school students spend like almost a whole rotation or a couple of years learning how to do this. It's a really cool skill. This is what doctors see under the microscope, so this is what their view is. And then they try to derive images or a vision like that. So the first problem I want to introduce is that these images are pretty hard to interpret. Like this is kind of a really blurry pink-purple image, and it's hard to get to there. The second and third problem that I'm going to talk about lies in the actual process of making these images. So this is when a doctor orders a biopsy, this is from start to finish how everything goes. The first step is to do tissue excision from the primary tumor site, or they might do something called a needle core biopsy. So they get like a needle, they'll poke the tumor with it, and they'll squeeze out the tissue that's in the center. They'll do a bunch of chemical processing steps, but the most importantly is they'll embed the tissue in wax. Once you have like this cube of wax with the tissue in the center, they'll slap it on like this deli slicing machine and they'll cut it up into really thin slices. Those slices go on microscope slides and they dunk them in inks. And this is a really critical stage because you have to choose what ink will stain the tissue. Because certain inks will stain for nuclei, certain stains or inks will stain for connective tissue. So this is a really important step. The go-to ink or the go-to stain is something called hemotoxin and eosin, H and E. The H stains for nuclei and the E stains for connective tissue. This is the go-to, this is the gold standard. It gives you enough information to usually make a diagnosis or some follow through with a clinical intervention. Second problem is the choice of stain. Third problem is when you choose a stain, you have to stain the whole tissue. So it's an all or nothing choice. If you choose H and E, all you'll see for the entire tissue is H and E. And this can be problematic if you're interested in not just nuclei and connective tissue, but if you're interested in quantifying proteins and things like that. And the third problem is that this whole process takes 1 to 4 weeks here in Canada. So in some cases, by the time you get your biopsy results back, your tumor or cancer will have already metastasized to a point where you need a new biopsy. It's kind of crazy. These are the different types of special inks or stains. Amyloid is a protein that's very prevalent in the brain for neurodegenerative diseases. There's different ones for muscular degenerative diseases and things like that. So for a single piece of tissue, you can only choose one of these. What if, like in a perfect world, we could basically go from this tissue block to getting a single slice of a tissue, and you're able to get all the stains, H and E, the ones for neurodegenerative diseases, and everything. So that way you can look at certain things if you're interested in that pathology, and you can look at other things if you're not. So the question I'm trying to answer is, can we study the microanatomy of tissue without having to stain it? It would be a really boring presentation if I didn't have something cool to show for that, at least. Um, and my research especially focuses on leveraging the optical properties of tissues. So if you think of tissues or molecules like a sponge for photons, whenever you pass light through that tissue or sponge, it'll soak up certain photons at certain wavelengths or at certain colors. So then when you look at after it's passed the tissue, you'll notice gaps in the spectra, like red will be gone or blue will be gone. And that's because the tissue's absorbed that specific wavelength. We take advantage of this because each absorption spectra is super different and very specific to the type of biomolecule. I'm interested in this DNA RNA range in the very deep UV. Some people are interested in oxy and deoxyhemoglobin for functional imaging, so looking at whether or not oxygen is present. You can get very, very specific down to like the single amino acid. You can differentiate proline from tryptophan, or you can go even farther in the wavelength to look in melanin and epidermal tissue. But I'm mostly interested here because the DNA RNA is what's the most useful, especially in the context of cancer. And the way we leverage absorption is by doing this novel technology called photon absorption remote sensing, or PARS. PARS is our own lab's technology. Like we invented it, we founded it, we have a patent, everything. The way it works is that we have some detection laser, so it's at a constant wavelength, and we're barraging tissues with it. So photons will hit the tissue, it'll bounce back. Certain parts of certain tissue will scatter more than others. So this is the first type of contrast that we're looking at. This is how we can differentiate some tissue from another. It's just using whether or not how much it scatters. We then introduce an excitation laser that comes in pulses. So it comes in like short little like boop, boop, boop. And the purpose of doing this is to raise the tissue's energy level. So we have this tissue, it jumps up energetically to a higher state. And once it's jumped up to a higher state, it has two different ways of coming back down, of relaxing back down. The first and most obvious or abundant way of coming back down, I shouldn't say obvious, the most common or like observable way is by radiating a photon. So when it comes back down to ground state from a very high energy state, it'll emit a photon. This will be a longer photon, like a longer wavelength photon than the one that excited it, but basically it'll fluoresce, it'll glow, kind of like how glow in the dark materials work. You have photons that hit it, it'll glow at some wavelength. This is very specific to each type of tissue. Another name that this goes by is called autofluorescence, auto meaning self, fluorescence meaning I'm radiating or glowing. The second and most important, at least to our context, is this portion. This is called the non-radiative relaxation pathway. Basically, now that the tissue is in a higher energy state, as soon as it absorbs the excitation photon, it'll heat up. And as a result of a rise in temperature, there'll be a volume expansion. With the rise in volume expansion, you'll see pressure differentials. It'll start vibrating. So it heats up, it starts vibrating. And that's when the photons from the detection laser that are constantly barraging it, they'll like scatter off in a slightly different way. So like because it's moving, the scattered photons will go somewhere else. And you can kind of use that as an indication for a type of contrast because absorption predominantly either results in a radiative relaxation or a non-radiative relaxation. So we're looking at both of these now. From scattering, we can see which areas scatter more. We can look at non-radiative, so we can see where, like, if it's not fluorescing, where's the energy going? And if it is fluorescing, what is it fluorescing at? So these are the three main contrasts, three main ways of differentiating some tissue from another. That's hopefully pretty interesting. This is my experimental setup. This is what I work with on a day-to-day. I'll go through it step by step. It's not as complicated as it looks. The first thing I want to highlight is our detection system. So we have a 405 nanometer laser at the bottom here. It basically just gets shot up right into an objective lens. And that's so that I could focus at the tissue. Think of it like a highway. You have photons going up, and then the scattered photons, as they come back, we have a special kind of mirror so that anything that's coming back gets bounced off into this photodiode. A photodiode, just like a type of camera for a single pixel. So it records the pixel value over time. And this is what it actually looks like. One of my main gripes with a lot of physics/engineering presentations is they don't show you what the signal looks like. So like you have this vague abstract idea in your head, but this is physically what I'm seeing. You see the brightness change for different areas. It goes up and down. The second part of the system I want to introduce is the excitation system. This is another laser and it's substantially bigger slash bulkier. And the reason being is that this is a super high power. It's a laser that goes from a really short deep UV wavelength all the way to far infrared. So it covers a very wide span of the electromagnetic spectrum, which is useful for if you want to move from different spectra to other spectra. So you can differentiate biomolecules more adequately. This is because I'm bad at physics. Basically, you shoot photons, and because it's like up here, it has to bing bong around into this beam expander. So it gets shot back up into here. It's mostly a logistical thing. If I could do it so that it just goes in nicely, I would. But because of like just the physical layout of the system, you have to do these fancy mirrors that like try to go up into it. This section is where kind of the magic happens, where some of the cool contrast stuff comes out. Basically, you have the excitation path that goes up into the tissue, gets absorbed by the tissue. It'll heat up and start vibrating, and it'll fluoresce basically at the same time. There's a very small like window in which you're capturing that. And because we're doing it in pulses, every pulse you'll see that event, and then it'll relax back down, ready for the next event. That's why it has to be in pulses. And then we collect the signal. So basically we have another objective. So because it's so like physically small happening, we have another objective that's collecting all of that photon data. It's sending it using a fiber optic cable into another expander here. Remember, there's the fluorescence and the non-radiative, so the radiative and the non-radiative. The non-radiative gets shot out. We have a special mirror that reflects at a 405 wavelength that lets everything else pass through. So reflect into this photodiode. You'll have the fluorescence do the same thing, or the radiative, go into it, and then go into that photodiode. It's just how we pick apart the two different signals because they're kind of coupled to each other. And the signals look like this. So this is exactly what I'm seeing. You introduce the absorption event at this pink line. It'll jump up in radiation, and it'll decay. Like the photon's brightness will decay. Likewise, when there's no excitation, you're seeing constant scattering, basically. Then you introduce excitation, and it starts heating up and vibrating. That's when you see scattering kind of dip down, and that's when you see a decrease in the brightness. This will have a different height or a different decay rate for different types of molecules. And the same thing with this. Sometimes it won't be as steep of a drop in brightness. It'll be like down to here, and it'll decay a little bit faster, or grow a little bit faster, or grow a little bit slower. And that's how you differentiate different molecules within a single sense of tissue. So that's the whole system. I hope that provided a little bit more context for how we take that physics and actually record it experimentally. That being said, did the experiment work? And I like to think it did. Oh, it's a pity because you can't see it too much. But these are the paintings that I was talking about. Maybe if I zoom in. Yeah, the contrast is terrible. But basically, you can see these blue regions, and if you look really closely, you can see these reddish regions. These reddish regions is the non-radiative contrast that I'm talking about. And it highlights mostly nuclei and DNA, like nuclear content. And these blue regions is connective tissue. That's kind of almost a one-to-one to the H&E stuff that I was talking about before. And as you kind of like sweep the tissue, you can see like, I forget, the projector's not doing it justice, but they're like these really gorgeous, stunning images of like these very bright fluorescent things that is giving you a lot of really rich information. And then this is the tail end. So when you're looking for cancer, you're looking at areas with a lot of nuclear content because cancers it's uncontrolled cell division. So you'll see a lot of cells just rapidly grow. And that's kind of when you're looking for areas that are crowded with red. Here's another type of tissue. Okay, you can kind of see this a little bit better. But basically, it worked. That's great. Did it really work? Were we able to grab clinically relevant or pathologically relevant features? Yeah, we were. So here are corresponding adipose cells here in this particular area of the tissue. You can look at down to the nucleolus. You can look at cell nuclei, individual ones. And it's, in my opinion, a lot easier to see on these images than it is to this. And this is really useful because now when you have two doctors looking at the same image, it's more likely that they'll come to the same conclusion. Because there's some ambiguity involved here. There's less ambiguity involved there because we're chemically deriving the contrast, whereas here we're kind of relying on inks and stains to hope to do that. Here's a really interesting area of this particular tissue. You can kind of see the sea of red nuclei. And there's like this little island of blue tissue. Oh, well, that's because It's necrotic tissue surrounded by tumor, probably. So this necrosis area, all like the cells have died, they've undergone a process called apoptosis. So their membranes dissolve and you have like these free-floating organelles, and you're surrounded with tumor that's probably what caused it to become necrotic tissue. And you could also investigate other types of different shapes of cells. So here I'm highlighting fibroblasts. They're kind of hard to see. And if you're not too used to looking at these images, they're kind of hard to identify, but they're really great looking. So that's like the bulk of my work. There's another PhD student at our lab that's graduated, Dr. Bakhtar. She's able to take one channel of these images and then using this AI technique. Remember when I was showing like those signals of like the different decay rates? You can cluster those by their shape. And when you have shapes that look very similar, it's likely they come from the same molecule. So you can do the same kind of labeling, but just using a single, so like this is the input image, and you're able to get that. And that's by doing clustering of the different shapes. So all these green ones are also nuclei. You have blue, that's still the connective tissue. You have this red, this is kidney tissue. So you can see the red is like glomerular tissue in this renal area and things like that. So this is a really exciting area as well because we're able to pull much richer information from basically the same set of data, which is promising for a lot of AI work because AI is only as good as the data you give it. So this is really cool and promising. This is two other PhDs in our lab that are hopefully graduating soon, Ben and James. They're working on doing the thing I was talking about, like in a perfect world, we can have all the stains. This is their input image. And they try to stain virtually using a type of deep learning network to go to H&E, 2D blue, and periodic acid shift, those different types of specialized stains that highlight more specific organelle or cellular data. And I'm hoping the video plays. Yeah, so this is skin tissue that we're looking at. You can kind of tell because there's like these columnar cells. This is the epidermis, and it gets more vascularized, or you'll start seeing blood vessels in the corner. So this is their work. They did this. I wasn't a part of this, but I think it's really, really interesting. They look at total absorption. So they look at the sum of the non-radiative and the radiative, and they look at something else called the quantum efficiency ratio. Basically, that tells you the gap between radiative and non-radiative, how big it is. So a larger gap would mean that it's more radiative, or sorry, more non-radiative. Smaller gap means radiative. Basically, it's another way of showing the energy absorption data. So again, you're able to get these, in my opinion, really stunning looking images. And most impressively, you're able to get to two different stains from one single image. So you can show that this special stain that perhaps, I think this highlights a different type of connective tissue, but you're able to show that without ever having to touch ink. Like these two images don't exist, which I think is crazy. Oh, sorry. Cool. And then when we ask the question, did it really work? We have to see if these images are actually useful to clinicians and doctors. These are two studies that Ben and James have done, the clinical studies that basically show doctors our image and a real image, and we ask them to make a diagnosis. And we see how often they disagree or how often they agree with each other on that set of images. And both of these show that we meet it. Like we're able to match the clinical gold standard, which is great. But in Ben's study, we're able to show that they actually do even better with our images. So on the PARS images, you can see that when two doctors look at the same image, there's better agreements in particular samples than there is with H&E. So they argue more with H&E, but they don't argue more with our images, which is really great and very, very promising. Now that I've done all that experiment, what's the point? Where do I go from here? I'm hoping to take advantage of this really rich set of data. You have these virtual stains of different types of like tissue. So you're highlighting very specific organelles. And I want to throw that into an AI model because I remember AI is only as good as the data. So I'm hoping that it'll be able to tell you what disease you have. It could highlight specific areas. This is the most diseased area. It could tell you what stain led to its prediction. So I thought H&E was the most useful here, or perhaps I thought a different special stain was the most useful here. I'm hoping it'll tell you about side effects. Like I've seen this most common in this cases. I'm hoping it might tell you even about prognosis. Like what's the outcome, the long-term outcome of this particular like tissue case. I'm also hoping to do what's called IHC or immunohistochemistry staining. So we're still talking pretty macro when we talk about cell and nuclei and organelle stuff, but I'm hoping to detect even lower, like down to the genetic level. So can we detect if there's a mutation in your tissue that'll manifest phenotypically? So it'll manifest as some alteration in the physical structure of the tissue. And if we can detect that and backtrace to a mutation. So these are different types of biomolecules that are very prevalent with different types of genetic mutations common in cancer. Uh, yeah, this is very exciting. That's also very exciting. And that's where like I'm hoping the rest of my thesis leads me, which will be fun. Thank you guys for listening very much. I love to take questions, comments. It's amazing.
Yeah. So the second laser, the high-powered laser, at varies in frequency, is the one that can detect the different like features by the molecule of molecules.
Yeah,
Okay, and so why is the first laser necessary? Why is it necessary?
yeah. So that goes into like laser physics. So this is the actual laser, and then this is what's called an amplifier. So basically, this actually excites at a wavelength of 1016 nanometers, which is like far in the IR range. And then this will take that 1016 and do some cool physics to basically output whatever wavelength you want. So the reason why this guy is necessary is because remember on the absorption spectra, different molecules absorb differently. So we want to be able to use this guy to move along the spectra so we can pinpoint specific molecules we're interested in.
Yeah, the first laser that like it gets the absorption back or it's like the reflection or another.
Yeah, there's a mirror here. Basically, the laser shoots here, mirror into here. It goes in a U process like this to basically shoot out.
That's,
Yeah, and the processing here is just to raise it energetically.
that's necessary for the second
Yes, to go between wavelengths.
Like this. A great presentation. I love the work so far. I just wanted to get some conceptual clarity
Sure.
on the difference that you derive. So there's no dye or anything that's being used, and it's pretty much we're working with light
Yeah.
and light waves. So with regards to the absorption, is that, or I mean to say, do cell like tumor cells?
Yeah.
They have a different absorption rate. Is that what we're working with here? Just clarify.
Yeah, so it's a bit of a nuanced question. And the reason why I say that is because superficially, when you look at cancer, you can characterize it by rapid cell division. So a bunch of cells start popping up. And when you have a bunch of cells popping up, that means you have more nuclear content, more DNA, because there's more cells. So that's the H&E way of going and diagnosing that. The reason why I say it's nuanced is because not only is that happening, you also have like genetic effects that are also altering the tissue in other ways. So we're hoping to, right now we're just capturing the first way, but we're also hoping to capture the second way by moving across wavelengths and pinpointing out where exactly those alterations are happening.
So you talked about wavelengths, right? And I could tell that maybe my assumption is that if we're talking about tumor cells, they have a different wavelength that's produced or
Yeah,
put up.
they're autofluorescence. It depends on the type of cancer. It depends on how that cancer is like manifesting itself, like how it's presenting. Some of them is really simple, but also like the thing about cancers is that you can get these things called teratomas. So it's different tissue types. So if you have a renal cancer, sometimes you'll see bone tissue pop out or skin tissue pop out. So in cases like that, absolutely you're right. You'll see like different fluorescence, but in other cases that might not be the case.
I have one last question. I'm really much into skin stuff.
Okay.
With regards to light, right? Does it lead to degradation of skin over time using this methodology of shooting light and getting all those, you're using mirrors to get all those different light spectrum, right?
Yeah.
That over time, I don't know, just side question.
Uh, Whether or not it's destructive matters very little to us because we're just hoping to pull out the image. Like these are already preserved wax samples, so if we destroy them, it's not too much of a loss. It does, you're right. Like you do have to lose some of the tissue to gain that contrast, but it's like very, very small. We're talking like pico level of like volume loss. Hopefully that answers. I'll go here first.
Order the first one for the AI use for the image. how much like hallucination or the kind of like imagination does use? Is it the generative where it could kind of best guess fill first thing, so giving more error?
Yeah.
Or would it be pretty one-to-one? I mean, what would be interesting?
It depends on what type of method you use. We have paired images, so we image using our method, and then we do staining with that exact one. So it's literally one to one. There's always a possibility of hallucinations with generative models. From the data that we've collected so far in some of our unpublished work, it's like almost identical. Like you're able to get pretty much 100%. I don't want to say 100, 90% like structural match.
And was it with the same tissue before and after stain? Or was it with
Yes.
different tissues
It was with paired
where
image.
it was never stained?
We've done both. So like the only way to get images that look the only way to get images like this is if you can only stain once, right? So we stain with this, and then we have to infer those other two.
So for this one, was it non-stained to begin with, then you did the light?
So we did the light thing very first,
Yeah.
and then we did H&E staining. So that's when we trained our models. And then with different set of images, we did the same thing, but for the other two stains. And then using those two trained models, we were able to make those two. And that's why like they're really good results.
My second question was, how does it change the timeline for when doctors would get their diagnosis back? Because you mentioned it takes up to 2 weeks or more.
1 to 4, yeah, it's
Yeah.
kind of crazy. A part of that's like logistical, like baked into the Canadian healthcare system, and the other part of it is just because it's a very slow process. Like you can imagine staining inks or processing and all that stuff. I can't give you a good answer as to how long our stuff would take. I can tell you it's faster, that's for sure, because you slap the image down, it images, and you get it back. We have one Ben, this guy, he's worked on a fast system, so his system takes like hours. The system I'm working with right now probably takes like around a day, something like that, to get images that look like these a day or two.
And would it have to be processed in your lab, or would there be opportunity for in the future being like on labs in the same hospital?
It's the same pre-processing, like the same fixation, dehydration, wax embedding. That's all the same. The imaging is where we kind of come in. So we don't do any special kind of pre-processing, usually.
Thank you.
Yeah. Sorry, I'll take a question from the sister side. Sorry.
Just out of curiosity, is there any like clinical or clinics here in Ontario that adopt this technique rather than the typical gold standard?
So the fun part about building medical technology is that you have to get cleared through like FDA stuff first. We haven't quite met that regulatory barrier. It's in the works. There's a startup called Lumisonics that works on like commercializing the technology, and they're doing some really, really cool stuff. It's quite rapid at the pace that they're going at. There's no clinics that are currently using it. We've imaged samples from Grand River Hospital just down the Ion. I've been talking to a pathologist that, or I'm hoping to image like lung samples pretty soon because they're genetically interesting. But no, nothing clinic yet. Yeah. Sorry.
Have you managed to apply this technique to DNA? Like, for example, some tissue might have genetic markers that say you're predisposed to developing cancer, for example.
Yeah. No, we haven't. It's for stuff like that. Predisposition is a really interesting problem because that happens like at almost, I think at like a somatic level. Like you're looking at mutations that are very different and might not necessarily, like you have to do sequencing for that, I'll say. And I don't think we're at a sequencing resolution. Yeah.
It seems to be like a disruptive technology compared to the previous way to do things. I imagine that it's like different in some areas, but it's rapidly moving towards the resolution, what types of PK. Like,
Yeah.
what currently is it like is sufficient compared to what's standard, like what ways?
Is it disruptive, like compared?
What currently is it deficient in? What kind of?
Oh, well, speed is a big one. The one stain per tissue is another really big thing. The chemical specificity is very, very huge. Staining, you have very coarse specificity, but with our stuff, we're looking at the absorption spectra, so it's very particular to that molecule. So I want to say there's almost like a world of a difference between the information offered in H&E and the information offered with our technology. To give you like the disruptive thing was I thought pretty cool to mention because H&E has gone back for like the past century. Like doctors have been looking under a microscope at cells for like forever. That was like the first thing that they tried. So having something like this could be a really interesting way of looking at microanatomy.
So who develop PARs and say like you're the only ones that have it, but like imagine other like they have their own slide based.
Yeah, well, PARS is like it's a physics principle, right? So anymore you can't patent MRI, you can't like patent PARS. We've built like a whole technology suite around it. So all the stuff I showed you is like ours. Our PI or like our head professor invented it at his lab back at Alberta. He kind of like pioneered the method, and then these guys did a lot of work to bring that method into like actuality, like into reality, like actually making it a commercial product, is what I would say. Yeah. Are we good on time? Okay.
Yes.
Okay.
Thank you very much for the nice presentation. Actually, one of my professors, one of the group that I worked before with, he worked in something using this laser-induced spectroscopy for
Yeah.
detecting like high metals in cancer cells. So is that method like quite different than this one or similar?
Yeah. So
I
spectroscopy.
mean, he did it in like 2019.
No. Spectroscopy works with a different set of contrasts. It mainly looks at what's fluorescent. So it'll look at what wavelengths the material or tissue is giving off. When you have high energy tissue, it relaxes in two ways. One of them is the radiative, and the other is a non-radiative. That's just like a fundamental like a biomolecule biophysical process. Spectroscopy looks at taking this and then looking at it across wavelengths. Uh, Ours, like our method, we take a look at that wavelength, we integrate everything underneath, and that's our pixel value. It could like it's definitely worthwhile and an interesting experiment to look at windows of that. Yeah.
Well, I love how you presented very top down.
Thank you.
Great. Second, you did mention that you're specifically using deep UV 405 nanometers to select for DNA and RNA.
Yep.
Could you translate parse later on to use different wavelengths to select for different areas of tissue to inspect?
Yeah, so our 405 is our detection laser. We're not actually exciting with that. Our main goal is to sweep across different wavelengths to excite at different absorptions. There's another PhD in our lab who looks at like looking at blood flow. He excites, I think, around like somewhere in the near IR range, so like 800, 700 nanometers. And then there's an interest to go into the IR range as well, like the far IR, like 1700, 1800, because UV is a little bit ionized. It's not ionizing compared to like X-rays or like CT stuff, but it's still like, it's not good.
What you're saying is like you can already extract a lot of info and infer a lot from this, but there's a much broader spectrum.
Yeah, there's a whole spectrum that's remaining to be explored. A lot of people do it. Like this isn't like we're not too special in that regard, but it'll be interesting to see because we're looking at the whole absorption event. We're looking at how photons radiate and how they shake and like vibrate non-radiatively. So looking at both of those things is kind of where we shine. Good.
Is that